Okay, good afternoon, everyone. So thank you very much for wonderful introduction. And Spinco and Shimazu, thank you very much for inviting me as a member of Avengers, Japanese Avengers. And I'm really honored to be here and speak to you today. So my title is Comprehensive Characterization and Impurity Profiling of Oligonucleotide. So today, before main topics, I would like to talk about myself and PeptiStar. So my academic background is organic chemistry and agricultural chemistry. And before PeptiStar, I worked for Shionogi. Shionogi is a Japanese middle-sized pharmaceutical company. And I have experience of working in CMC department, and the manufacturing strategic department, and the corporate strategic department. And the PeptiStar project was started in 2016, and I was a leader of a PeptiStar project. And in 2019, I left Shionogi, and I was committed to the growth of PeptiStar. And in terms of India, 10 years ago, I came to India several times, New Delhi and Chandigarh for cost-saving projects. And I established good relationship with business partners. And after relationship, I was invited to a wedding ceremony in India. It's really amazing, and it's totally different from a Japanese one. But I'm sorry, I'm not here to talk about the impression of an Indian ceremony. So let's move on to the next one. And PeptiStar is a peptide and oligonucleotide CDMO, and also PDC, peptide drug conjugation. And it was founded in 2017, and the operation was started in 2019. Now, as you can see, our employees are about 70, and the number of projects, it means from discovery to our most advanced product is a phase 3 compounds. And the background of foundation is before PeptiStar, there was no CDMO, which covers discovery to commercial in Japan. So PeptiDream is a startup company in Japan, and Shionogi and Sekisui Chemical decided to establish new CDMO. And also we focus on the manufacturing technology, and our location is very good access from Osaka Airport and Shinkansen Station. So we are always welcome to your visit. So PeptiStar has 3 main buildings, and headquarters and R&D buildings, and building 1 and building 2. And building 1 and building 2 are GMP facilities, and our capacity is approximately 100 or 200 kg per year. And also we have many kinds of synthesizer for peptide and oligonucleotide. And also peptide and oligonucleotide, basically their compounds are not crystallized. So we have to consider the process of purification and lyophilization. So we have many kinds of reverse phase column and ion exchange column and a lyophilizer. And we have experience manufacturing many kinds of peptide and oligonucleotide. So long-chain peptide and a cyclic peptide and a branched peptide and a cyclic homodimer peptide and PDC. And in addition to peptide, and we have experience of many kinds of oligonucleotide. So long-chain, aptamers, and guide RNA, it is for CRISPR-Cas9 technology. And the DNA, RNA, and siRNA, and PMO. So in addition to the manufacturing technology and the manufacturing equipment, so analytical equipment is really, really important in this area. So Shimazu supported PeptiStar from the beginning of our business. So our ratio of Shimazu equipment is about 80%. So India is 40%, so we are trying to increase the percentage of Shimazu analytical equipment in India. So the reason why PeptiStar select Shimazu, they are very user-friendly, and they are strong support for maintenance and good communication. And when I worked for Shionogi, so I used Shimazu equipment, but design is no good at that time. But currently, the designs are getting sharp and very nice. Okay, this is the main topic today. And this is an overview of CMC development oligo from drug discovery and preclinical and clinical. So in PeptiStar, after receiving order by customer, we try to synthesize the compounds by our standard method. So after that, we checked the quality. So main impurities are shortamers and longomers and conversion. On the other hand, so analysis of impurity is very important for optimization of synthesis and the purification process. If we don't have the information, so we cannot go move forward next step. So after optimization, and we have to move to scale up and quality control. However, analysis of impurity is very, very difficult. It is not easy. For researcher, it's very annoying. So as a CEO, I have to consider the pains for researchers. So what are the pains for researchers? So there are many kinds of impurities in API, and several impurities are very close and occasionally same retention time. So as a result, it takes time and effort for analysis and identification of impurities. So in order to ease the burden of researchers, so Shimazu and PeptiStar started joint research in 2021. So from PeptiStar, we provided our customer view and a customer request. And after receiving our request, Shimazu developed a new software based on their technological capabilities. And after 2 years, we finally released new software last year. This software is amazing and provides strong support for impurity analysis of oligonucleotide. So what can Insight Biologics do at present? So Insight Biologics has 5 solutions. Number 1 is to estimate impurities in same molecular way. Number 2 is to estimate impurities in same peak. Number 3, enhance MS quantitativeness. And number 4, obtain key information on one screen. Number 5, complicated structure can be exported to ChemDAO. This is number 1 solution. So as you can see, UV chromatogram just showed the retention time. So it is impossible to estimate impurities without MS information. So next step, we check MS for getting information of molecular weight. So after getting molecular weight information, we can just estimate the impurities roughly. However, it is not enough. For example, M-1 mer. So right side, so image of target material. However, M-1 mer, there are several candidates. So next step, we have to check MS spectrum, fragment information. After getting this information, finally, we can identify and estimate target impurities. So left side information, we can get this kind of information by hardware. However, right side, so without software, we have to calculate by ourselves, something like Excel file. However, Insight Biologics automatically propose impurities. And number 2 solution is to estimate impurities in same peak. So sometimes we face difficulties that several peaks in the same peak, several impurities in same peak. So we check mass information. So basically, there are a certain pattern in impurities. So inside biologics, each peak to each impurities. B1 impurities peak and a B2 impurity peaks. And next one, I would like to talk about quantitative analysis, because a little problem is MS. It's a little problem, quantitative analysis. So because in oligo, there are several peaks of multiple charged iron in impurity. So as you can see, if pick up the different peak, occasionally the peaks area in MS are different from one in the UV chromatogram. So currently, current system is that convert to MS chromatogram based on one charged iron. However, inside biologics, so the convert to MS chromatogram is based on all charged iron. So this data is more reliable. I'll show you that one example. This is impurity peaks very close to target material. So we would like to separate each impurities after identifying and estimating the impurities, PS, conversion type, and shortmers. However, if the data is not reliable, we cannot trust the data. On the other hand, as I mentioned, the inside biologics provide reliable data. So this is very useful for optimization of the synthesis and purification. And solution number 4. So inside biologics enable us analyze impurities on one screen. So we don't have to change application on one screen. So after getting old, so if we change the application and change the screen, so we lost current position. However, everybody check key information on one screen. So UV chromatogram, MS chromatogram, and decomposition spectrum, and MS spectrum, MS spectrum. In addition to these key information, this software proposed visual check of possibility and the proposed impurities candidate. This is really a useful function for researcher. Without this information, so researcher get lost the way. And this is image of a UV chromatogram. So after making this software, we can create impurity mapping. So next step, we can optimize the reaction condition and the purification condition. And number 5, so for researcher, basically they have to make a report to upper management or give a presentation. However, the structure of oligonucleotides are very complicated. Sometimes it happens a mistake. However, inside biological, researcher input a sequence, so this software provides the structure automatically. This is very useful function. And this is summary. So Shimazu and PeptiStar developed a new innovative software, and this is already released. And as I mentioned, so this software has 5 solutions. And Shimazu and PeptiStar and SPINCO will never stop updating this software and hardware. So I hope my presentation works for your investigation, next step in oligonucleotide. Thank you very much.